Welcome everyone. Welcome to the second part of the study design and quantitative thinking module. Let's quickly go over what we saw in the first part. In the first part, we looked at replication and interspersion. We discussed several points related to replication, including what is an independent replicate and how do we figure out whether we are replicating at the correct scale, and we also looked at examples of pseudo replication. Let's continue on talking about replicates, and let's focus on an issue that invariably we have to engage with, which is how many replicates, that is sample size. How large a sample size do I need? You may have heard estimates like you should sample 5% of the population, or you should have a sample size of 30, but actually there isn't a clear number. It really depends. What are the factors that sample size depends on? First, it depends on the variability in your system. If there is a lot of variability, then you need a larger sample size so that you can cover this variability and you get a representative sample, and so you can generalize from sample to population. Sample size also depends on the magnitude of the effect you want to detect. That is, do you want to detect a difference of 3% or a difference of 30%? Let's take an example to understand this. So, I had given you this example of logging and recruitment in the previous component, in the previous session. So, let us say I am interested in understanding whether juvenile densities, so this is of a bird species that I suspect is affected by logging, that is the felling of trees in a forest, and what I would like to do is see whether the number of juveniles or the abundance of juveniles who represent the future reproductive component of the population. So, I am interested in whether the number of juveniles varies between logged and unlogged forests. Now, if the difference between the two types of forests, if the difference between the two types of forests is small, so 3% difference, then if I have a comparatively small sample size, I wouldn't be able to estimate the mean number of juveniles in unlogged and logged. I wouldn't be able to estimate it precisely, which means I will have large error bars, that is large confidence intervals, that is high uncertainty, and I wouldn't be able to detect an effect of 3%. I would need to increase sample size so that now I have been able to estimate juvenile abundance in logged and unlogged patches. I have been able to estimate this precisely. Small error bars, high confidence, low uncertainty. So, now I will be able to detect this small difference of 3%. On the other hand, if I want to detect a large effect, so a large effect of 30% for example, then even with a comparatively small sample size, even if I do not estimate these mean number of juveniles in the two types of forests, even if I do not estimate them precisely, I will still be able to detect this difference. So, sample size depends on how large an effect or how much of a slope or how strong a correlation you want to detect. The question is, how do I figure out? Do I want to detect a 3%? Do I want to detect a 30%? And for this, I have to ask myself, what is biologically meaningful? So, for example, in this example of recruitment of a bird species, we know that mortality in the lifespan of many bird species, mortality is highest in the juvenile stages. So, a large proportion of juveniles actually die, and adult mortality rate is usually much lower. So, if mortality rate of juveniles is high, then a small increase because of logging, an increase of 2 or 3% may not matter biologically. On the other hand, 30% is likely to make a big impact on the population. So, depending on the biology of your system, you will have to think about the natural history and the biology of your system. You can arrive at what kind of an effect size, meaning what kind of a difference or slope or correlation, what is meaningful and what you would like your study design to allow you to be able to detect. Okay. Apart from variability and magnitude of the effect, a third important factor that affects sample size is how many parameters you wish to estimate. That is, how many different relationships do you want to estimate in your study. So, apart from looking at the effect of logging on juvenile abundance, maybe you are also looking at the effect of resource availability, predator density, etc. And the more number of such relationships you want to estimate, the larger your sample size has to be, so that each of these can be estimated with good precision. Okay. Now, once you have decided how large a sample size you want, that is how many replicates you want, how do you select replicates from among the sampling units available for you to sample and measure? This is where randomization comes in. We select replicates at random, and we do so. We randomize because we want to reduce bias and improve generalization. That is, we want to get a representative sample of the population, because only then can we generalize from sample to population. If our sampling protocol gives us samples only from a subset of the population, then we cannot generalize to the whole population. However, it is important to remember that randomizing is actually quite tough. So, for example, in this barking deer example, suppose I wanted to estimate male and female body size, and I want to sample 30 males in the population and 30 females in the population. So, I could decide that every day I go out and measure the first 5 males and first 5 females that I encounter, and then measure them. The problem with such a sampling protocol, which is often called haphazard, it is called haphazard, where you have an arbitrary protocol like the one I told you just now. The problem with that is that it is possible that I do not get an unbiased sample. I might sample the most active individuals. If I choose the first 5 that I encounter, I might just end up choosing the most active individuals, which means I would be generalizing towards the active subset of the barking deer population, not to the whole population. So, I need to figure out a sampling strategy that will give me an unbiased sample, that will allow me to select males and females at random. What do we mean by at random? It means that every sampling unit has an equal chance of being selected. So, in this example, every male has an equal chance of being selected, or every female in the population has an equal chance of being selected. Let's look at another example. So, let's say my objective is to estimate tree girth of a particular species of tree in a forest. So, this is a large area, large forest with thousands of trees, thousands of individuals of this particular tree species, and I want to know what is the mean tree girth. This might help me with biomass measures and so on later on. So, now I want to select individuals at random to measure. Remember, selecting at random means every tree in this forest of this species has an equal chance of being selected. So, this is the strategy I come up with. So, I say okay, I have the coordinates of the forest, let me choose lat long at random, let me then go to that spot, find the closest tree and measure it. So, I am choosing coordinates, latitudes and longitudes, I am choosing coordinates at random, going to that spot chosen at random, finding the closest tree and measuring its girth. So, I would like you to spend a few seconds to think about whether this strategy is, this sampling strategy is going to allow me to choose trees at random. As it happens, no, and the reason is as follows. In any forest, trees are unlikely to be uniformly dispersed. There might be local tree falls or some other small disturbance for a variety of even stochastic reasons. You are going to find that trees in a forest are not uniformly dispersed. So, this means if my randomly selected pair of coordinates falls say here in an area where tree density is high. In such a case, these trees, these individuals in areas of high tree density are much less likely to be sampled, each individual, than if my coordinate were to fall in an area of low tree density, in which case the trees in such an area of low tree density are much more likely to be selected at random. That is, the area within which an individual is likely to be selected at random is much larger for a tree in a low density area than for a tree in a high density area. So, all individuals do not have the same chance of being selected, and therefore, I am not carrying out random selection. If I could actually mark each tree, then I can give numbers to each tree and then select numbers at random. That would be perfect. So, that would allow me to choose individuals at random, but that is often not possible, because imagine in a forest with thousands of trees, I would not be able to a priori mark each tree. I will have to find some other protocol. So, for example, maybe I can make grid cells that are typically likely to enclose only one tree and then choose grid cells at random, discarding ones without any trees in there. Or maybe I have larger grid cells and then I select trees within such a grid cell in proportion to their density. So, I will have to think through potential sources of bias and then come up with a sampling strategy that lets me select individuals at random. I would like to really emphasize this point that randomizing is tough. So, let us take another example. Let us say I am interested in measuring the behavior of fish in a stream, and I want to measure the behavior of fish in groups of different sizes. Now, I can't follow the behavior of all individuals in a group. So, I need to select a few individuals whose behavior I can measure. So, the question is, how do I sample individuals in a group at random? So, remember again, at random means all individuals in a group should have an equal chance of being selected. So, maybe a sampling protocol that I come up with is that I select a point on the periphery of the group at random. I also select a number at random, that is, which individual should I choose and monitor. So, say I come up with this location at random and I come up with the number number 3 at random. So, then I move inwards towards the center and say I will measure the third individual that I encounter as I go in a straight line from the periphery towards the center. The problem is, even though I am choosing a location on the periphery at random, even though I am choosing an individual from that location to select and measure at random, still the protocol that I have just described to you still makes it more likely for fish in more central areas to be selected than fish in more peripheral areas. So, I have to figure out a different sampling protocol that will allow fish to be selected to have the same probability of being selected for measurement. Okay. The next point I would like to make is that even if we can figure out randomization protocols that are very good, that are very unbiased, true randomization is not always best. Why is this? So, typically our sample sizes are moderate or comparatively small. So, for example, if I want to measure the girths of trees in a forest of a particular species, I might choose 20 or 30 or 50. I typically won't choose thousands of trees. Now, in a very large area with a very large sample, with a very large population, if I try to choose a few trees at random, just by chance I could get peculiar arrangements, like for example, sampling in this case where I have indicated selecting 5 individual trees at random. So, here is an example where I have selected 5 individual trees at random. 4 of the 5 are in a high tree density area. So, now if I estimate tree girth, it is unlikely to be representative of the whole population. It is likely to represent tree girth of individuals in high density areas, perhaps of individuals experiencing high competition. So, I cannot generalize to the whole population, but to the subset that experiences high competition. This is the same idea as in when you have a coin, even a fair coin, if you flip it very few times. So, if you flip a coin 8 times, there is a good chance you can get all 8 being heads, even a fair coin. On the other hand, if you flip that coin 800 times, you are very unlikely to get 800 heads in a row. So, especially with small samples, you can get if you select individuals completely at random, you can get peculiar arrangements like this. Let us look at one other example. Let us say I am interested in the effect of grazing by large herbivores on plant communities. So, I am interested in grazing by say large antelope or large deer on plant communities. So, if we take the example of large antelope and plant communities in a grassland, let us say I want to carry out an experiment in which I compare plant communities in areas that are fenced. So, grazing by large antelope has been excluded. So, areas that are fenced versus control areas where antelope can move freely through these areas. So, I want to see how plant communities differ between fenced areas and control areas. So, now fencing is quite expensive and maintaining fences requires a lot of effort. So, typically I might have only a moderate sample size, maybe only 10 or 15 fenced exclosures that exclude these large antelope and 10 or 15 control areas. Now, suppose I follow true randomization, that is in a grassland. Here this rectangle shown here, I select points at random and assign them to either fenced or control. So, here I have shown you an example with 5 fenced plots and 5 control plots. So, just by chance I get this particular set of locations for fenced and control plots. Now, if I measure plant communities in fenced and control plots, and if I find a difference between fenced and control plots, one inference could be that excluding antelope has resulted in this difference in plant communities. So, effect of grazing by antelope has a certain difference, has a certain effect on plant communities. However, take a moment and look at the arrangement of fenced and control plots in this grassland. You might argue that if there is, say, a soil moisture gradient, maybe it is wetter at this end of the grassland and drier at this end of grassland, then it is possible that the effect I have found in plant communities, the difference that I have found in plant communities is not because of grazing, but it is because this area is wetter and this area is drier. So, just by chance we can get these if we locate our sampling points at completely at random, just by chance we can get these peculiar arrangements. What is the way out? If there is a known gradient, so for example in the tree example, I have a good biological reason to believe that tree density represents the level of competition experienced by individual trees, and therefore the girth of trees can be affected by tree density. So, if there is a gradient that I suspect, then what I can do is I can stratify based on the gradient. So, I can grid up the area and stratify grids based on tree density and then randomly sample within these grid cells. Suppose there is no known gradient, but I suspect that there is heterogeneity. So, for example, in the fencing and grazing examples, suppose I suspect that there is heterogeneity in the grassland, but I am not sure what is causing that heterogeneity, then I can block. That is, I can choose locations and in that area I can create both fenced exclosures to keep out grazers and close by I can place control plots where grazers have access, and then compare plant communities. So, here what I am doing is, if there is some underlying environmental feature like high soil moisture, both treatment and control experience it, and in another area if there is some other environmental feature like low soil moisture, both treatment and control experience it. So, in this way I get rid of any confounding factors. So, a third point to mention about why we typically do not use true randomization or complete randomization is that if our sample sizes are moderate or comparatively small, if we choose individuals or choose locations completely at random, we may not get good coverage of the population. So, for example, in the grassland case, where I was interested in looking at the effect of grazing on plant communities, if I choose locations at random, maybe certain areas of the grassland never get plant communities there, never get sampled. So, another way to carry out sampling is to bring in a systematic component. So, we could have large grids, grid up the grassland into large grids, and in each grid cell we could decide depending on the sample size available to us, we could decide that we can put in a certain number of samples. Then we could find locations at random and there have blocks of treatment and control. So, we have a systematic component to ensure that blocks fall in different areas of the grassland, and therefore, we have good coverage, and then we use a block design to make sure that treatment and control are fairly close by, and therefore, we can get rid of any other confounding or correlating factor. Okay. Let us continue on this theme of blocking and talk about locating sampling units. So, let us go back to the example of looking at the recruitment of a particular bird species, that is how many juveniles are successfully produced during a breeding attempt. So, how does juvenile density vary between logged and unlogged forests? So, because in this landscape we can have all other kinds of confounding features like differences in predators or differences in resource levels and so on, we might want to have a kind of paired or block design where we compare juvenile density in pairs of logged versus unlogged plots. So, let us say in this landscape, this rectangle, these yellow patches are unlogged forests and the surrounding area is logged. So, what we do is we randomly select a patch within the unlogged forest, a plot within the unlogged forest, and then close by in the logged forest we establish a plot for comparison. And by locating them fairly close to each other, we make sure that any other confounding features are shared, and therefore, we use this block design to take care to incorporate or account for confounding factors. Now, suppose we find at the end of the study that recruitment, so juvenile density is very similar between logged and unlogged patches. One inference is that logging does not seem to have a large effect on recruitment. However, we also need to check whether these pairs of plots were independent from each other. That is, imagine that once juveniles leave the nest, suppose they disperse within a certain radius of the nest, then it is possible that from these unlogged patches you have individuals moving out into the surrounding area shown by these red circles here. And similarly, from the logged forest also you have individuals moving within a certain radius. So, now the measurements between pairs of logged and unlogged patches are not really independent. Juvenile density in the unlogged plot can affect, or the unlogged, this patch here can affect densities in the logged plot and vice versa. So, while creating blocks for your treatments, controls, or natural variation, this does not have to be only for experiments, this could also be for an observational study. So, in this case, in this logged unlogged patch case, we are using natural variation and we are placing our plots. So, this is not an experiment, this is an observational study. So, whether it is the grassland fencing grazing study or this logging and recruitment study, we are trying to establish blocks to take care of underlying heterogeneity. However, we have to think carefully about the distance between pairs of plots. And that is the last point I would like to make here, which is while we want to carry out procedures like stratifying, blocking and so on, we also have to think of the appropriate spatial scale and the independence of sampling units when deciding which are the sampling units that we want to establish and measure. So, to summarize this second part of the study design component, we have now gone through replication, interspersion And now a third key element of study design, which is randomization.